Hey there, guys. Okay, for this quick movie, we're just going to show you our lesson that we did for gel electrophoresis and how we can make a DNA fingerprint. So here we go. So first of all, our goal was how is DNA used to identify individuals, and the process in when you're trying to identify individuals called gel electrophoresis. And this is also known as a DNA fingerprint. But if you notice, fingerprint is in quotes, okay? It has nothing to do with the fingerprints or your fingerprints. The reason it's called a DNA fingerprint is because it's unique. DNA fingerprint markings are unique for each person, just like the prints on your finger. So what do we use it for? Well, we use it for CSIing, identifying suspects in a crime. We also use it in Maury, solving paternity mysteries. And awesomely, we can figure out which organisms are most closely related, evolutionary relationships, like human and chimps are closely related, and chickens with T-Rexes. Ooh. Okay, where do we get human DNA from? Well, we need human cells, and so you can do a swab for a cheek cell, you can do blood, skin, hair, tissue, saliva, other. And as long as you have a cell, one cell, you can get a DNA sample from someone. Remember, stuff like hair doesn't really have DNA, so you need either skin or part of the root, which carries DNA. And interesting, red blood cells do not carry DNA because they don't have a nucleus. Now, how do we isolate the DNA? That means how do we get it by itself if you got all these cheek cells? Well, the first thing we use is a machine called the centrifuge. And the centrifuge sort of breaks up all the cells. You stick the sample in the centrifuge, spins like crazy. And then after a few chemicals and stuff, the cells parts sort of separate according to their density. And so it turns out DNA is really, really light. So it floats to the top. You get a pipette and you grab it. And then you're going to pipette it into what are called gel wells. That's hard to say. Gel wells, gel wells, gel wells. The gel is sort of like jello, and it's like a porous jello that if you stick the DNA in there, it'll move through the gel. It'll like swim through the gel, and then you can sort of make your DNA fingerprint marks. So it works a little something like this. The picture on the left, upper left, is a DNA fingerprint. The bottom left is the machine that's used, the gel electrophoresis machine. The blue stuff in the machine is the gel. Okay, in that picture in the bottom left. In the upper, the red line here is going to represent a DNA sample. And so what the first thing they do to make a DNA fingerprint is you take someone's DNA sample and you cut it. It'll cut into pieces like that. Now, since each person is different, each person will be cut in different places. And the thing we use to cut, I'll tell you in a second, but we cut the DNA and then we stick it in the gel. We turn the machine on, and the pieces move according to their size. So the pieces that are biggest, they sort of move the slowest. And the super, super tiny DNA pieces move really, really fast. So if someone had a DNA sample that was like 4 big pieces, they would have 4 lines close to each other. If someone had a DNA sample that was 1 big piece and 1 little piece, then when you put them in the machine, that would look like 2 stripes. 1 stripe far away from the other one. Why far away? Because the little ones move a lot faster than the big ones. Let me show you. If you're looking at a DNA sample, the ones at the top of this, near the question mark, are the smallest. They move the furthest. The ones at the bottom of this picture, close to the guy's thumb, those are the fattest pieces because they're closest to the wells, and the wells is where you put in your DNA sample. Now, how do we cut the DNA? Well, the DNA is cut by some awesome thing we discovered bacteria have called restriction enzymes. Restriction enzymes are these little enzymes that can actually break up DNA in specific spots. And now bacteria use this as like a defense. So if a virus attacks a bacteria, they actually can use the restriction enzyme to like slice up the viral DNA, which is awesome. So we discovered that we can use these restriction enzymes as like our own little personal scissors. and we learned how to splice genes. That means we can cut and paste genes using these enzymes. So, for example, if this represents a gene and we have a specific enzyme that looks for AATT and cuts between AATT, then we get something like this. We get two pieces, a small piece of DNA and a large piece of DNA. If you notice in the sample, there's no more AATT. It doesn't matter that there's an AT at the end there. It has to find all four letters. And restriction enzymes come in different flavors. There's GATTC, there's CCGG, there's a bunch of different ones. So we have to tell you the restriction enzyme on the reagents or a lab so you know where to cut. This is what the machine looks like. You take the DNA sample from a test tube, you crack it up with the restriction enzymes, you stick it in the wells, you turn the machine on, and the pieces move.
Are added to the wells at
Whoa.
the top.
Whoa, forget that. I hope that worked. The DNA pieces will move because of an electric current, okay? And then you look at the stripes, and whatever has the most stripes is the match. So remember in class we played Who's Your Daddy? And we did these DNA samples, and we cut them based on AATT. And if you notice, they're all a bunch of different sizes. And then we stuck them in a gel. And when you stick them in a gel, they sort of look like this. And you compare Jimmy, who's the kid, with the three fathers. The most matches wins, which means Harry Potter is your daddy. Okay? That's how you do a DNA fingerprint.